Good day, everybody, and welcome. I'm Jack Van Horn from the University of Virginia, and please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. It's my honor and privilege to welcome our speaker today, Dr. Tor Wager, the Diana L. Taylor Distinguished Professor in Neuroscience at Dartmouth College in Hanover, New Hampshire. His completed a Bachelor of Arts degree in music from 1992 to 1996, and completed his PhD in psychology in 2003 from the University of Michigan. He has held faculty positions at Columbia University, at the University of Colorado in Boulder, and since 2019 has been at Dartmouth College up in New England. Under the direction of Professor Wager, the Cognitive and Affective Neuroscience Lab studies the neuropsychology and neurophysiology of the affective processes involved in pain, emotion, stress, and empathy, and how they're shaped by cognitive and social influences. His lab is particularly interested in how thoughts, beliefs, and expectations affect the brain and the body, and they use a range of techniques, including psychophysiology, electroencephalography, pharmacology, as well as neuroimaging to understand those processes. Relevant to the brain and data science, his lab is also dedicated to the development of new computational models for the analysis and synthesis of neuroimaging data, especially fMRI, and to work to promote the open sharing of tools and scientific data. Additionally, Dr. Wager is also no stranger to providing online educational lectures on neuroscientific data analytic approaches. He has many honors, and he is funded currently through a number of NIH R01s. He has had funding from the National Science Foundation, the Army Research Institute, Templeton Foundation, and several other grant awarding bodies. Again, numerous awards from the National Science Foundation, the American Psychonomic Society, amongst others. And he also has 2 patents that he is the holder of on a variety of research projects. His topic today is going to be the neuroimaging of pain and affect, generalizable models for prediction and explanation. And as always, we are streaming this lecture live and for recording via YouTube. And if you are watching on YouTube, thank you so much for joining us. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Wager via the chat feature in their Zoom sessions. I will synthesize these questions and ask them on your behalf during the last 10 minutes or so of Dr. Wager's lecture. And with that, welcome, Tor. We are looking very much forward to your lecture.
Great. Thank you so much for inviting me. It's really a pleasure to be here and to be able to share some of our work with you and some ideas. I'm going to go back to the main reason to do neuroimaging in the first place, which is that neuroimaging, the promise is that it'll play a unique role in this chain from molecules to cells to systems to human behavior and disease. And since the start of neuroimaging research in humans about 20 years ago, I think we all thought that this is going to take off and change the world. I think it is changing the world, but it's also taken longer than expected. And there have been a few bumps along the way. Many of those bumps have made it into media in exaggerated ways. So here in this cover article from New Scientist a few years ago, it says the vast majority of brain research is now drowning in uncertainty. And there have been a number of critiques that have been important, including critiques of effect sizes and how we estimate them, critiques of power and sample size in neuroimaging, critiques about reproducibility and how we make inferences about different pieces of the brain, especially here around Howey threshold. And most recently this year, critiques about reliability of task fMRI measures. And as you see at the bottom there, since this is from 2010, Max Coulthart, but the history of these kinds of critiques about what neuroimaging can tell us about the mind go way back to the very earliest imaging studies as well. And if you boil these critiques down, there are the following things, which I was just talking about, effect size, power and false positives, reliability, inferences about the mind, and inferences about translation. They do extend these critiques beyond brain imaging itself. So here's an example of an important critique of gene-by-environment interactions. And the field of genetics was really very early in jumping on this idea that we have, too many false positives, which stem from sort of too many tests. And, these are very broad critiques, of course, that many of us know about, why most published research findings are false. And neuroimaging has even been compared with phrenology, which, as I'll show you, is an uncharitable comparison, because there's no situation under which phrenology can yield externally valid predictions and inferences about the mind or personality or individual differences. But with neuroimaging, there is a lot of promise that we can indeed construct measures of task states, of mental states, and even clinical phenomena that are externally valid and reproducible and reliable. But the central claim I'll make in this paper is to suggest to you that we've been doing it wrong. We need a paradigm shift. And that paradigm shift is from constructing maps, which are local estimates of different bits of brain and what each bits of the brain is doing, to integrated models, which are a model in general is a system where you specify what the parts of the system are and how they interact with one another. And applied to brain imaging, a model will be specifying how different pieces of the brain and different brain measures combine together and interact to predict an outcome that we care about clinically or behaviorally or in terms of performance. And central to this idea is that if we can construct stable models in the brain of which brain features or measures we're integrating in which way, then that leads us to strong and falsifiable predictions. Much of the kind that Einstein, for example, made based on his theories back in 1915, which were tested years later by Eddington. So Einstein predicted that starlight the light from the sun would lens around the moon when during the eclipse. And that test of this, the first empirical test of this, was really what put Einstein and relativity on the map. So that's a goal for us. And I'll also claim that we need a biomarker development framework or a model development framework where we iteratively optimize brain patterns and features. We construct models and markers of outcomes that we care about. We validate those prospectively and test their generalizability, specificity, and other measurement properties. We characterize their robustness, their boundary conditions. What do they respond to and not respond to? Under which conditions do they work and not work? And to the degree that we have valid and validated models, then we can test them and we can start to use them to understand what the structure of the mind is like, as represented at least in the brain. So central to this idea is the idea of contract validation, understanding what something measures. And this takes a lot of work. This is one of the reasons that we're really at very early stages of developing these models. As a counterpoint to some of the critiques, I'll point you to, this is a review that we wrote a few years ago now. It's a snapshot of translational studies. And here we had almost 500 studies and more than 500 predictive maps that all use machine learning or pattern recognition techniques to do various things in various disorders. And you can see the breakdown across disorders here. There's more things coming out every day. This is a rapidly growing field. And on the right, what you can see is in all of these different areas of mental and brain health, there are models that seem to, in this case, predict who's got a disorder versus controls with high accuracy. And on the y-axis is you see the accuracy there, close to 100%. Each of these dots is a study, and the size of the circle represents the sample size. And the larger circles are large studies, even up to and over 1,000 participants. So we've come a long way. And these kinds of models are not perfect, but they're being combined with increasingly large data sets through massive data sharing and data and model sharing efforts. And that's going to be a theme of today's talk as well. So this is a courtesy of the UK Biobank. We go from a small study to a big study to now the UK Biobank study is the largest study, I believe, currently being collected with 100,000 individuals. So we're really moving towards population neuroscience. And here's a small table of a number of really important large-scale efforts where data are being collected and shared with communities to analyze them and discover new things. One example is this, recent effort by Vince Calhoun's group. They call it Neuromark, and one of the important features of this is that it's a system of classifiers based on independent component analysis. They fed in lots of data from several of these large samples. So they have 12,000 people in this paper overall, and they're predicting things like diagnostic category. All right, so very much different from phrenology of old. I'm going to turn to an example from our lab and use that to illustrate the biomarker development and testing process and what it can do for us. So traditional brain mapping is really the assessment or the conduction of a number of separate tests. So we'll look at a little bit of the brain, or voxel, and say, well, okay, which psychological task stimuli or task states are predicting activity in that voxel? And then you do it again for the next voxel, and the next one, and the next one, for 100,000 voxels or something. And what you get is a map, a collection of different local effects. But it does little to help you understand how it works, or even whether you can predict a new instance of that task state. A predictive model flips the equation around. And so now multiple brain voxels or other features, they could be connectivity features or graph, topological measures or anything else. But the idea here, where I'm going to illustrate with simple linear patterns across voxels, is that these brain measures are allowed to combine together to jointly predict the outcome. And once you have developed a predictive model, you can test it prospectively in various ways. So you beat on it, test its properties. We can apply the models that I'm going to share with you to new samples, new individuals from different scanners and different populations. And you can use those to predict continuous outcomes. We'll talk about predicting how much pain are you feeling now, or categorical outcomes. Are you a patient or a control? Are you unconscious or conscious? Are you feeling pain or are you not feeling pain? And so forth. So a simple version of how this looks schematically, and I think that there are many algorithms for doing this. I think the algorithms are interesting, but not necessarily the key thing and not the focus here. That we'll take a training sample, we'll take a group of individuals, and here we're going to apply multiple levels of painful stimuli, from barely painful for that person to about as painful as they can tolerate. So there's 4 levels per person. And then we're going to use essentially a giant regression model to identify a linear pattern that gives us the best weights for combining brain regions to predict pain experience. So that's what I'm going to call a signature map, which is a precisely specified pattern that we can validate. And where sort of machine learning comes into it is how good that model is depends on how you construct the algorithm and how you sample data. So 2 key ideas that many of you know about are cross-validation to develop estimates, and some techniques for stabilizing those models so that you don't get a bunch of garbage, essentially. So you have sort of stable interpretable maps. I'll come back to that more at the end. One of the first models we developed was called the neurologic pain signature, because we want a name that we can reuse and refer to. And the journal editor suggested it, and we didn't want to offend the journal editor, so that name's back. And this pain predictive pattern includes many areas that we know and love in pain research and beyond. And the key thing here is that there is a precisely specified pattern within each region, and we're integrating across regions. And all of those areas are really sensible in terms of mapping onto invasive research in humans and animals. And so there's backing from neuroscience studies. We can do many things with this model then on new data applied to different individual subjects. So we can take a series like this where people get a threat cue and then a painful or warm stimulus, and then they recall pain. So we have 3 different sort of phases of the trial. And we can apply the pain signature to calculate a single response for every trial or event. And so we can take those trials like this for a person. We can use those to predict how much pain they're feeling. So now we're looking at the pain-related responses. And you can see this is one representative subject. the actual sort of sequence of temperatures they have here, and then the predicted sequence. Here you can see the model trained to predict how much pain a person's feeling. Each line is a person, and each dot is an average over several trials within that individual person of different intensities. And we can take those and we can resort those trials into the different types of events, and we can do then a test, again, in new individuals, so it's unbiased, which is more painful. Is it the heat, the anticipation period, or the pain recall period? And we can then, and what you see here is that this pain signature I just showed you responds with increasing stimulus intensity when you're actually getting heat at the moment. It does not respond at all to anticipation or pain recall, judging pain afterwards. And that's been consistent across many studies now in our group and others. So here, in a out of subject classification, we can take a new person, and we're 100% accurate in the sample, at deciding which are the pain conditions. Then we can apply this to new test data, where we take something from a person from a different scanner, we apply that signature, and what that means is we just take the pattern weights as a template, take a data image coming in, and we can calculate the dot product. And there's some other metrics as well, but that's the main one that we focused on. And that boils the map down into a single value, which in this case is the predicted pain, the pattern response or pattern expression. And now we can see here that if we apply the NPS to this new study of multiple stimulus intensities, it responds with an increasing function, just like pain itself actually does. And we can tell which is more painful out of conditions where people get a 48 degree or 47 degree stimulus, for example, with 100% accuracy per person in a forced choice test. Here's another application to a new subject from a different scanner. So they get 46, 47, 48 degree noxious heat, low, medium, and high. And we can, each line is a subject. And then we can turn that into a classifier. That's a continuous readout. We turn it into a classifier by saying, which is more painful, the high or the low? And if we were to make a guess for each person in this sample, we would get it right about 96% of the time. And that's been a benchmark across studies for this particular pattern. So this is really a way forward. What are some of the advantages? Well, for one thing, we've reduced the analysis flexibility. I fixed the pattern. And so instead of testing 100,000 things and trying to correct for multiple comparisons across them, if I'm trying to validate that signature response, I just do one test. And that leads to strong inference or falsifiable tests. Number two, it capitalizes on distributed brain codes, and that can increase effect sizes and reliability dramatically relative to looking at the sort of standard paradigm of single regions or single boxes. And third, it opens the way for model sharing. So I can share this with you. I'll come back to this at the end. And you can test it for yourself. And you can share data with us if you want to, and we can test it. And that helps us to establish what its boundary conditions are. What is the thing actually tracking? And validate the construct that it's actually measuring. So here's a little bit about reliability. One of the meta-analyses that just came out this year is really a broad critique of task fMRI as being unreliable and perhaps too unreliable for to serve as biomarkers. That's sort of a claim. And this was a response that was just accepted in Psychological Science, so it's coming out soon. And what you see here is the reliability of pain ratings themselves across 8 studies, 8 dots a study, which are excellent. And you see the reliability of the NPS pain signature, which is also in the excellent range. And here we actually have quite strong reliability for even some individual regions responding to pain, like the anterior cingulate and insula, which is not always the case actually, but the distributed pattern is quite reliable. Here's some other examples. This is from, the one on the left is from Pete Gennaros' lab, a large population health study predicting essentially physiological measures of cardiovascular disease risk. And they have a short-term retest reliability here that's also in the excellent range. And you can see there each dot is a person. And what's happening here is it's the first half of the session and the second half. So it's a short-term retest. I'm happy to talk more about reliability as well. On the right, you see 2 examples of clinical classification. So the top one comes from really a landmark effort by Drysdale and colleagues in Connor Liston's lab that develops biotypes for depression. And in independent samples, they can classify depressed versus control patients with high accuracy. That's an external validation. And they can identify which of those biotypes they are accurately. And on the bottom, you see a task state classification, faces versus places. So it's just a demonstration that using the same data from the HCP, Human Connectome Project, that Elliott et al. Used in their analysis, we can actually identify a distributed pattern that is highly reliable at identifying what state people are in. And as I said, the critiques of science recently have gone really beyond fMRI and neuroscience even themselves to all kinds of fields. And psychology has been one of the main ones. And so what you see here is a sort of one of the landmark papers in this area. And they point out that analysis flexibility is a huge problem. Researchers have many, many decisions to make, decisions about whether to stop collecting data or collect more, which outliers or data points to exclude, which measures to analyze, which covariates to use, which types of pre-processing and modeling and correction. I'm a fan, for example, of Josh Karp's paper, where there's thousands and thousands of different ways to analyze the same data set, and they give you different answers, right? And of course, which findings count as support for a hypothesis that I'm going to come back to in a second. So the term, p-hacking sums up a lot of things. It sums up this idea of, iteratively making analysis decisions or changing your interpretation in order to increase the likelihood of finding something that you want to see, consciously or unconsciously. So this is a powerful force. And I'm just going to show you a recent paper that we did with Wani Wu, my former student, who's now a professor at SKKU in South Korea. And this really illustrates the idea that analysis flexibility in the standard brain mapping paradigm is a killer. So here you've got, this is a real data example from social rejection study that we did a few years ago. And what you see is, a pattern of activity in response to being rejected by another person. And so typically you might conclude the anterior cingulate cortex is activated, there's a hotspot, it survives correction for multiple comparisons. But if you look at the unthresholded patterns next to it, you can see there's a lot of information there that's lost in that simple summary. Now we do a replication. And we do the same contrast in a different sample. And again, we get the same conclusion. The anterior cingulate's activated. Wow, this is a direct replication. But if you look at the pattern, what's the signal distribution, it doesn't look so clear that it's actually a replication. And so typically what we do is we'll pick a coordinate from each of those and we'll report that coordinate and we say, oh, we replicated it. But how close does that coordinate have to be? How similar does it have to be from one study to the next to count as a replication, right? Those coordinates are really arbitrary. In this case here, there are no voxels above threshold in common. So if you looked at the same voxels, there's not a replication. The peaks are 4 centimeters apart. And for reference, the amygdala is only a centimeter and a half big or something. So 4 centimeters is a huge distance in the brain. And if you look at the patterns of activity, those patterns are uncorrelated. So what Wani and his team did was look at 135 studies that claim replication. And most of those studies don't provide enough information to actually assess replication. They just sort of talk about the same brain regions. Of those that do, the average spatial distance between the original and the replicated peaks is, over 2 centimeters. So it's also very, very high. And so this is a real problem because it's going to prevent us from aggregating results. And what I'm trying to convince you of here is that this idea of picking the peak coordinate and then, comparing it or trying to replicate that sort of general area is essentially bankrupt. It's just too imprecise. Another well-known study that came out, which is also sort of quite critical, was by Rotem Botvinnik-Nezer and a group of people called the Narps study. And what they did is essentially generate a prediction market and had a number of teams test a specific set of hypotheses about a task, and which the hypotheses, what they are, is not so important here. But the main thing that you can see is that these different teams analyzed the same data, and they concluded different things, right? The decisions they made about these hypotheses, yes or no, are in many cases not very consistent. So 70 independent teams chose 70 different pipelines. And so that was sort of one of the taglines of this paper. But if we just look a little bit deeper, there's actually more interesting stuff going on. Now, this figure from the paper shows the correlations between the spatial maps. And the reds are positive correlations, so that's good. And the entries are teams, and the reds and greens are hypotheses, which are sort of vary, right? But even though the threshold, the yes or no decisions vary, the actual maps are very similar for a large proportion of the teams. So the patterns that the different groups are seeing are highly correlated, except for this little bit over here, where they think they conclude that some people kind of did the analysis wrong or made some mistakes, right? So there's another cluster. Okay, so the patterns are actually much more stable than this decision where you apply a very stringent threshold and you look at the tiny peaks that survive and you go, how similar are those, right? So that's not very stable, but the patterns are. Okay, and I'll show you more examples too of effect size and reliability. But now I'm going to go to the third thing here, which is this idea that every brain area has diverse populations of neurons in it. So when we measure a voxel, we're measuring something that is heterogeneous. There are many different populations. And there's a very strong literature, and so you see some of the papers here that's arguing that those individual neurons, and by extension individual voxels, are not how information is encoded or represented in the brain. In fact, what's a key for representing information is the pattern across the neurons and across specific ensembles of neurons within each region. So our job in imaging should be to identify brain signals that track those ensembles, that track specific ensembles, and test whether they're tracking information about one kind of a set of ensembles or one population code or another one. And that's what I think this model building approach that we've been talking about is doing. From this review, which I really like, Averbeck and Pouget, they say, as in any good democracy, individual neurons count for little. It's population activity that matters. So highly relevant in US politics at this moment in time today. So what are the principles? Well, cells with different functions are intermixed in local areas. Individual neurons are often noisy and they're not closely linked to behavior. And even when they are, many studies demonstrate that it's actually the pattern across all the neurons, not just the sort of most responsive or most selective ones that's important. And the kinds of psychological functions like pain or sadness or anger or working memory performance, all of these things, or sustained attention, are not implemented in isolated brain regions. They're implemented in circuits. And those circuits involve specific ensembles in local areas, and they span multiple areas of the brain. So we have to characterize the circuit. And here are just some examples from the neuroscience, which is generating amazing data right now, amazing revolution in what we understand about circuits. And here, these are all examples where you can tag, genetically tag and stimulate a particular ensemble of neurons. And when you tag that ensemble, then you get different behaviors. So in this study, for example, you know what they're tagging is an ensemble related to encoding a fear memory, and they can reactivate it and then reproduce the behavior. But it's a very specific ensemble. Okay. All right, so now I'm going to move on to thinking more about what are the properties and benefits of these models. And I want to introduce this section by making the following claim that I hope you might find controversial, which is that the kinds of predictive models that I showed you can serve goals of both prediction and inference. And therefore, they are really a way forward for many of the answers in neuroscience that we want. So this is a tale of two doctors. And these two doctors are going to look at the same brain pattern, but want different things out of it, same brain model. So Dr. A is a translational neuroscientist, and she wants to study the brain to develop biomarkers for human disease. And why do we need those? She wants mechanistic targets for what's happening in the brain as she's doing treatments or testing treatments. Dr. B is a basic scientist, and he says, I just want to understand the brain. I just want to understand the brain representation, how it codes information. So Dr. A is interested in biomarkers, which are physiological, objectively measured processes that indicate some mental experience. And why she's interested in them is because the hidden causes of diseases and mental health disorders that she's interested in are hidden. They are not directly observable. Take pain, for example. We can't see pain. We often can't trust other indicators. And in many cases, the pain doesn't exist yet. It's a predisposition. So if you're going to develop a prognostic biomarker, right, you need to know who's going to get pain later, or a predictive biomarker, who's going to respond to a treatment that you can't see. So these are really hidden mechanisms. And the main value of this brain imaging, I think in this case, is to develop those systems models, not to recapitulate pain, but as mechanistic targets for diagnosis and intervention. So for example, is this baby in pain? Well, we don't know. We can't observe the pain. We can only see the behavior. And of course, babies can't tell us. This is a high stakes question because in fact, even today, babies get painful procedures because it's commonly assumed that they don't feel pain the same way that adults do. And there's a long history of that. But there's excellent work in neuroscience from Mariah Fitzgerald, Rebecca Slater, and others, Simon Beggs, that really suggest that they really do show all the neural and behavioral characteristics of being in pain. Now, if you take a step back for a second, how would we know? What are the underlying mechanisms driving the pain? Well, for thousands of years, theories like this one persisted, that there's 4 kinds of humors: yellow bile, black bile, blood, and phlegm, and they mix in different ways, right? So this affords a whole one whole set of treatments based on what you think the underlying causal mechanism is. And of course, in modern scientific view, we don't think that's true anymore. But what is it in the brain that's in the body and the periphery that's creating pain? Okay, let's turn back to Dr. B now. So Dr. B is interested in representations. A representation, I'll define here, a brain representation, as a causal obligatory physical basis for a mental experience or information processing structure. So it's something where if you can activate it in the brain, you always get pain, let's say. And if you turn it off, you never get pain. So Dr. B might be interested in this question. Well, what's the representation of pain? And there have been a number of claims that have gotten a lot of attention that when you observe other people in pain, that activates a shared sensory representation. It activates your pain system, and that helps you empathize. So Dr. B wants to know, is that the case? Are there shared representations? And so I will suggest to you that there is really common ground between prediction and explanation. Although there are distinct goals, if you think about them, there's the intersection is really important. So what is prediction without explanation? Well, it's a black box. In a black box, you don't know when you can trust it. When is it being unfair? What's driving its behavior? So it's not very useful. And there's a big debate now about interpretability in all kinds of machine learning models. And if you have explanation without prediction, it is just a story. It's an unfalsifiable explanation because it doesn't make any strong predictions. And so you don't know if that explanation is right or if it's useful, or if it's true. So in the intersection is explanation that's supported by testable, falsifiable predictions. So both Dr. A and Dr. B here are really trying to measure brain signals related to an underlying construct. It could be pain or anger or stress or intelligence or memory, etc. And if they're measuring constructs, there's a common set of empirical criteria that we ought to be interested in. And those are whether the model is testable in new individuals whether it's sensitive and specific to the construct that you're trying to measure. So if pain is a construct, it should sensitive means it responds when it is pain. Specific means it doesn't respond when it's not pain. It ought to be robust, generalizable across different types or instances of pain. It ought to be interpretable in terms of the circuits and other literature and converging evidence from neuroscience. And we ought to be able to actually use it, and test it across labs. So Dr. A might be interested in, if you have a generalizable model for pain, the claim is that it's going to respond across multiple types, thermal, mechanical, visceral. And so Dr. A says, what is this thing really measuring, right? We don't know. Is it tracking all kinds of pain? Is it tracking only pain? And Dr. B might say, well, which parts of the brain are important? What's the key brain representation? Is that shared across social pain or observing other people in pain as well? So let's look at generalizability for a second. There's this idea that there are gradients of generalization of multiple kinds. So for any brain model, you might be interested in what it tracks. Is it only a very specific stimulus condition, one type of pain in one spot? Is it a class of similar states? Is it very diverse exemplars of all kinds of exemplars of the construct like pain? You might be interested in which populations, in whom. Is this model only applicable to one person in one sort of moment? And that's what most of the MBPA or predictive models from vision, for example, and other areas are designed to do. They're only supposed to apply to one person. But all the models I'll show you are designed to generalize to other groups of people, maybe new individuals from the same sample or population, maybe multiple samples of different characteristics, or maybe even heterogeneous populations. And you might be interested in which contexts does this model predict the outcome it's intended to predict, let's say pain. A very narrow context with the exact same cognitive and stimulus conditions, a similar context, or a very, very broad set of contexts. Is it robust across many of them? So we can test, if we have a model that we can apply to new individuals and new studies, which we do, we can test this in various ways. So this is just one example of that same pain signature being tested on a data set with 20 different studies from around the world, 600 people. So there are relatively diverse populations, at least compared to most studies. And it's diverse in terms of the types of pain that's being assessed. Electrical, heat, laser, mechanical, visceral, which is actually a rectal balloon in this case. So multiple types of evoked pain. And what you see here is each of the rows is one of the studies, or the 20 studies. And a shift to the right means the study is the NPS, the pain signature, is responding to pain. And the small dots are individual participants. So you can see all the individual participants. And what you can see is that it responds in the right direction for the vast majority of participants, 95%. And the effect size is massive. For reference, Russ Poldrick published a really nice paper called Scanning the Horizon in 2017. And the broad claim for our purposes is that if you look in areas where you see a real signal, there's a real signal and you're measuring how big it is, but it's an isolated area, effect size about 0.5, which is moderate effect size, moderate reproducibility and power. And here the effect size is much, much larger. So you can really see this in individual people. Let's take a look at specificity. I'm going to go back to this idea about observed pain and self-pain, experience pain. And it really does matter. So one of my favorite sort of foils here, David Brooks, although like many of the things he's written, he wrote this article called The Archipelago of Pain. And what it claimed is that self-pain, physical pain, and looking looking at other people in pain or experiencing social pain all look the same in the brain. And because they all look the same in the brain, then we really ought to treat emotional distress differently when it comes to policy. And indeed, should we treat emotional harm like physical harm then, right? And indeed, there are published papers and a lot of data suggesting that at the sort of overall level, these things really do look similar. They all activate the anterocingulate cortex. They activate the anteroinsular cortex. So here's a pattern-based study. So what are the population codes show us? Well, here we have somatic pain and vicarious pain. So looking at other people in pain. And we have 2 locations for each, arm and foot. And we have 3 pre-selected intensities designed to create a variation in the range of somatic pain and a variation in the range of vicarious pain. So how bad is it? And what we can see is these are their pain ratings, that they're matched across both. There's really a nice distribution that's similar in pain ratings, and people think both of these things are bad, somatic pain, vicarious pain. Okay, so now we can ask, well, what happens with our pain signature? Does it respond to vicarious pain? The answer is no, it doesn't. It tracks pain when it's self-pain. Those are the warm colors across 2 body sites. It doesn't mean we can't discriminate body sites too, but this pattern works for both body sites to a large degree. So it's a really nice dose response function, but there's no response to looking at somebody else in pain. So at the pattern level, this is quite different. And in this paper, we demonstrate this in many, many ways, essentially, beyond what I'm showing you here. And we can take the same data and we can ask, is there a signature in the brain or a pattern that tracks vicarious pain, the intensity of vicarious pain? Again, all these are new individual subjects. So they're all sort of unbiased tests that you're seeing here in the graphs. And the answer is yes. And it generalizes across hand and foot and maps stimulus intensity very nicely, but there's no response to self-pain. So this pattern is called separate modifiability, and it's one of the strongest criteria that I know of for process separability. We've got 2 patterns that respond to 2 things. We can do this across the brain. We can do this with individualized patterns. We can do it with selected areas of the brain that are supposed to respond in common, like the anterior cingulate cortex, and we get the same answer. We can also replicate this with different paradigms. So here's another study where women came into the scanner and their male partner was outside the scanner. So they experienced pain for themselves, and they were watching their partner's arm through a mirror, which was always present, so the visual simulation is constant, but then they were cued that their partner was going to experience pain. And so there were some periods where they knew that their partner was getting pain, even though there was no visual cues. And we can look at the neurologic pain signature for self-pain, vicarious pain signature, and we again see this separate modifiability process. So, and you can see the individual subjects here as well as the bar graph in the group. The NPS responds only to self-pain, not to pain on the other, but the vicarious pain signature responds only to pain on the other, not to pain on the self. So it replicates that pattern in the new study. And the thing about specificity is that it's an open-ended process. You can always think of something that maybe that looks like pain, but it's not pain, right? Maybe it's breathlessness, or maybe it's something else, or maybe it's sounds, or maybe it's some, you can keep going, which is great, but we have to, of course, test that across studies. So it's an open-ended process. So we can assess specificity as well as generalizability on new cohorts. And so for this demonstration, I'm going to show you a data set that Phil Craigle collected together from multiple studies. And here there's 270 participants, and a third of them get pain. And the pain is divided up into 3 types of pain: thermal, visceral, and mechanical. And there's 2 studies of each type of pain and 15 subjects. And there's 3 studies of cognitive control: working memory, response selection, response competition, 2 studies of each, 15 subjects. And there's 3 types of negative emotion: from pictures, from social cues, and from auditory cues, sounds, and 2 studies of each type. So it's a systematically sampled set. And the point of Phil's paper, I'm not going to tell you about in this talk, but I encourage you to read it if you're interested in generalizability, because the point is that you can identify generalizable representations using this systematic set that are not shared across other domains. And you can test their generalizability. But for now, I'm going to stick with the pain signature. I'm just going to show you the NPS responses across all of these data sets. And again, the dots are individual subjects, the bars are studies. So you can see that there's a threshold at which almost all of the pain subjects respond. And in very few of the other conditions, maybe one of the auditory studies there, most of the studies, they don't respond. The NPS, or I should say the NPS does not respond, right? You can try all these things yourself, by the way. So we built a whole system of tools if you're interested in this. And you can use our CanLab core tools on canlab.github.io, and you can try this, with 2 lines of code and reproduce it. So overall, we've been actively sharing data and models with people across laboratories, and it's been really fun because I've sort of gone around and given talks and said, well, maybe you have something that looks like pain, but it isn't pain. It's an interesting test case. So let's test it out. And by sharing these data across these different cohorts, we've been able to characterize what this measure responds to and what it doesn't respond to. Of course, there's more to go, but this is a snapshot so far. It does respond to multiple types of evoked pain, and to some degree, things like our somatic distress, like breathlessness, aversive taste, and painful sounds. Although they don't seem to track with the intensity, they don't track the intensity in the same way. And it doesn't respond to a number of tests, and some of which we've discussed, aversive images, arousing, salient, horrible things, looking at the person who rejected you, so social rejection cues, looking at other people in pain, anticipating pain, and other conditions, including some somatic ones, potentially. Okay, so now there's this idea that when you have gone through this process and validated a model in all these ways, the more you know about the model, the more useful it is in then testing interesting test cases and making some inferences about the mind. So this is where these hard-won inferences about the mind come in at the end, right, of this process. And so you can ask, is this baby in pain? Well, again, we never know because we can't observe that baby's internal state. But what we can do is apply the NPS as a validated measure to infant data after appropriately normalizing them. So Eugene Duff put a lot of work into trying to match the brains and apply the signature. And what you can see here is a validation in adults. It responds. This is the sort of pro-pain nociceptive part of the NPS. It's responding, in adults. There's an anti-pain system part, which is this sort of default mode in visual cortex and things that are deactivated with pain. It does not activate. And so that's a validation that this system is working in this study for adults across stimulus intensities. And then applying it to 2 studies in infants, where again, that pro-pain NPS is increasing across stimulus intensities. So infants are encoding that. And that's one piece of the puzzle, among others, that suggests that yes, infants have very similar neurological responses to pain as well as similar kinds of behaviors. Okay, so I'll talk a little bit about interpretability and model sharing before I wrap up. So interpretability is really important, and we've been thinking a lot about how to do this. And so I want to point you to this paper that came out recently. This is from Wani Wu's group, carrying on a sort of a elaborate and detailed program of research. And in this paper, we really thought about different kinds of ways of interpreting and validating models. And so there's a feature level assessment, which is some human readable representation of which voxels or which features contribute to that pattern. So how do you reconstruct a sensible map? And there's a model level assessment asking things like, does this track confounds or is it independent of confound? Could be head movement and many others, right? Is it generalizable, et cetera? And biology level assessment, which is, which means that you have to take the features of the model and interpret them in relation to past literature and animal studies. So do you get sensible pathways that match what you'd see in animals and can validate externally? I think all those things are really important. Okay, so then, we can develop experiments, and there's this whole progress. I'm not going to walk you through all of this, but there's a whole process that we outline in this paper of developing provisional models and then doing a lot of the hard work to say, does it perform well? Is it independent of confounds like head movement or medication usage or something like that if it's a clinical biomarker? And that involves testing in new data. And then in starting to interpret the model features. Do they make sense? Are they in the brain? Are they in places where we can validate them? Are they stable and reproducible features? And then, is the model generalizable? Is it biologically plausible? So some award-winning models in the brain have turned out to track activity in the ventricles where there's no real signal. So that they're a problem, or track head movement essentially in fancy ways, right? And if we fail these tests, we have to sort of go back and re-engineer better models. And then we can start to understand, how it performs on new data more broadly, and so forth. So this is some simple ideas. We can take these resting state networks as just one way of characterizing any of these models, right? And we can break them down into the loadings on these large scale networks. So here's a signature that tracks emotional experience. And you can see which networks it loads positively on: ventral attention and visual networks. And inside the circle, by the way, there's negative correlations with frontoparietal, dorsal attention, somatomotor networks. But if you look at our pain signature, that's what we've been talking about, the NPS, it loads positively on ventral attention and somatomotor networks, a very different pattern. And if we look at the vicarious pain signature, it also shows a very distinct pattern of network loadings. So they're really different large-scale systems that we're tracking. And here's some code if you're interested in that. Another kind of interpretation is we can ask how much of the brain and in which systems do you need to accurately make a prediction about pain or about emotion or whatever your outcome is. And so in this demonstration, we have a comparison between the best single region, which is way down here. It doesn't work very well at predicting pain. And the whole brain model is way up here at the top end. And what you can see is a single region doesn't do very well. As you add voxels, that's you moving to the right, you end up getting more and more accurate, and it levels off here at some point. And these lines are when you run the prediction with only an individual network, only the somatomotor network, or only the voxels in the ventral attention network. And you can see that no model really does as well as the whole brain. And Bogdan Petri has a preprint from my lab that really goes further and characterizes this in a lot of detail. I think it's a great paper, but I'm not going to talk about it here. We can also take a model like this, which is across the brain, and we can break it down into its local information content. So this is a predictive model from another study that we developed based on several hundred people. And then we can go back and say, well, this is a pain predictive model. Well, what about the thalamus? Is the signal, are the contributions different in different parts of the thalamus? And here you see that they are actually, and this is a beautiful thalamic atlas by Morel and the Morel Atlas, which they shared with us. And you can see here, what I put stars over is all of the thalamic zones that really are known to convey nociceptive and pain information from animal studies and human invasive studies. And so you can see the larger the wedge there, the greater the pattern weight there, the greater the contribution in the model to pain. And we can break it down into thalamic regions and other brainstem regions that are really important in animal models, but that are very seldom discussed in human models. Okay. And now I'm just going to wrap up by talking about model sharing and going back to this idea of the snapshot of translational neuroscience. Well, we have a problem here because we have 500 studies and we have 500 different models. And we don't really need, 100 models of Alzheimer's disease and 100 models of PTSD, which are all different and hard to compare. We have what my old colleague Walter Michel called the toothbrush problem, which is nobody wants to use somebody else's. And in fact, in this review, we characterized the models at the time, and there are a small handful that have gone from single sample to prospective validation in a new sample, actually being tested in a brand new sample, unbiased. And there's a few models that are named here. You can see this, ORCID, STAN, so there's some Alzheimer's models. SPARE AD is an Alzheimer's, PDRP is a Parkinson's disease related pattern with by David Eidelberg's group. And so, as you march into the more and more data and more replications across laboratories, there are very few of those named models. But that's a really important thing. And we have to share models and test them. So to that end, we have started sharing all of our models. I got inspired by the idea of a model zoo in AI. A lot of the deep neural network models, for example, are shareable and reusable, and it's great. So we have a website here and a GitHub repository where we have shared multiple things. All of the multivariate predictive models, including now the NPS that I showed you and others. So you can download them and try them out for yourself. And there's a growing family of them. Meta-analysis maps from meta-analyses over the years that you can apply to new studies, brain parcellations, and some of these are from our lab, and many of them are from other labs that agreed to share these data too. I'd be happy to host them as well, because I think the more focal points for sharing we have, the better. So in sum, I have talked to you about the idea that we need a paradigm shift from isolated brain maps to integrated models that predict outcomes. I've used pain prediction as an example of how we can test this model across many conditions and studies, and share both data and models. And I've talked about some of its properties and benefits, including large effect size, high reliability, high reproducibility of effects, high specificity to a construct, in this case pain, and high generalizability across multiple types of evoked pain. And then finally, I've talked about the idea of sharing these models and working together, which is really the fun part of science. And I'm just going to end by saying thank you to my current lab members and my former lab members, many of whom are collaborating still and have contributed a tremendous amount, and to our funding agencies over the years. And thank you very much for your attention.
Tor, thank you so much. That is really an impressive body of work to try to understand the brain's response to pain and the role of cognition, emotion, with the goal of trying to find out what can be generalizable, what can be explainable, and how you can leverage computational models in order to give you some insights. I want to invite our biomedical data science seminar series fellows to contribute any questions they have for Dr. Wager via the Zoom chat feature. And while we're waiting for those to accumulate, I wanted to get your thoughts. You presented a number of different studies, some of which were kind of mega analyses, that is just very large numbers of subjects that you actually have the data for and can reprocess, versus some of the other studies that you looked at, which were meta-analyses, where you're drawing from what people have reported in the literature. And just in my own experience, and perhaps this is yours as well, the studies which tend to be very critical are those which are the meta-analytic ones. They always seem to be the ones that kind of show everybody what they're doing wrong. And I'm curious about your comments about that in the context of reliability, and whether or not the choices of processing, which are going to be very heterogeneous when you're doing a meta-analysis of the literature, are those different from what you're doing in a very large sample mega-analysis like the UK Biobank? And if you have any thoughts on the distinction there, and whether or not that's an important factor in some of the heterogeneity that we're seeing.
Yeah, thanks, Jack. I think I spent probably 15 years, we're still doing coordinate-based meta-analyses, so summaries from published papers, to characterize what's consistent in the literature. I think those things are really useful. And like I said, we're still doing some, but those coordinate summaries, like a paper and a table, is a really impoverished sort of picture. And so we've really tried to move, I've gotten excited about, towards this sort of mega analysis approach, where we're getting, the full image data, the full set of effect sizes, because you can do so much more with it. And ideally, even the individual person level maps, because then you can really test, for individual by individual, how often would we make a correct prediction? You can look at spatial similarity across, brain patterns and data sets, and you can test, the replicability across subjects and make population inference in that way. So I like the meta-analysis. I've become a real fan of the, sort of the integrated data sharing mega-analysis approach. And I think it can be used to make very critical points, but it can, but it can also just be used to generate new hypotheses and data and models.
Are there any dangers with problems of like publication bias in meta-analytic assessments? Where really people are only publishing those things which are statistically significant. So we're leaving out a possible bunch of null results that could inform some of our meta-analytic statements.
Yeah, no, right. I think there's lots of issues. One that comes to mind that you sort of highlighted is, if let's say you're summarizing published effect sizes from papers, right? And every fear study does a region of interest analysis in the amygdala, and they always find it because the threshold is very liberal, right? And so what do? Do you include everything? That's what we've sort of done, and then you find the amygdala is really important. But is it really the strongest signal in the brain related to fear? No, it's not even close. Right, and there's some new work that actually shows that. Or do you leave those out and say we're not going to publish ROI analyses, but then you've excluded all the studies where people tested principled hypotheses, right? And now you're like, so it's really a hard problem. The idea of bias in sampling bias and selection going in is a really difficult problem. If we share data and we get the full maps, that actually really helps, I think, in that regard.
We're at the top of the hour here, but I want to ask one more question. There's a bunch of them coming in, which I'm happy to share with you offline, Dr. Wager. But because of time, I want to get to this last one. Really, what is the role? I mean, you presented a bunch of work that really looks like it has a lot of data science in it. There's a lot of computational modeling. You mentioned machine learning, deep learning, support vector machines, a number of different techniques. What role do you think that card-carrying data scientists have to play in order to help identify robust and reliable patterns that might be useful as biomarkers in things like pain and the brain?
I think that data science is super important. I mean, it's like basically essential skill set then to be able to do these things. But what I'll also say is that what I think the most successful sort of sweet spot is somebody with the skills, the data science skills, the technical skills, the mathematical skills and computational skills, but also a real interest in the brain, in understanding how to translate the sort of technical bits, right, into what are people going to understand? What does the field need to know? What do we need? What level can we frame this or describe this to really make it understandable for the audience, which is neuroscientists and psychologists and the public. So I think there is this sort of real intersection of interests, right, that's going to characterize very successful people in this area.
Well, with that, I want to thank Dr. Tor Wager from Dartmouth College for a fantastic tour de force on discussing of how to characterize the brain's response to pain, how to generate models that can be both explanatory, predictive, and useful, and for sharing them all. I think that's wonderful. And so thank you again, Dr. Wager. I really appreciate it. I want to thank everybody who joined us, and I want to wish everybody a very good weekend. Take care, everyone. Thank you.
Thank you. I'm happy to answer some things over email too. If you send me the chat, and I can respond to some of the questions.
Fantastic. We'll do that. Take care, everybody.